Right, it's absolutely great to be here. I'm Pete Hollingsworth, Deputy Keeper and Director of Science at the Royal Botanic Garden, Edinburgh, and the main focus of my work is on plants. And I do that because despite Paul saying that they don't deal with the numbers very well, and they're really difficult to barcode, they really are critically important structural components of terrestrial ecosystems. And despite that importance, there are many, many cases when the inability to identify a plant impedes our understanding of ecological processes, conservation planning, or plant use. So it's a really, it's my sales pitch for plants. So in the UK, we don't have a formal barcoding network. It's rather a loose assemblage of researchers that come together and form up for collaborations on individual projects. When I got this through, it reminded me of my school reports, sort of modest performance, could do better. A little bit embarrassing, but in part that reflects a difficulty in persuading the UK research councils to fund the infrastructure around library construction. It also in part reflects the fact that a lot of the UK barcoding activities are on non-UK samples, so a lot of our records will appear elsewhere. The flora is complete, the vascular plant flora is complete, and the bryophytes are almost complete as well. So just whizzing through a few projects. This is something that's recently been funded, coordinated by the Natural History Museum, Synthesis Plus. This is a network project across European natural history organisations, and a key bit of the current iteration is a project looking at DNA sequencing on demand. And this is really building on the fact that there are great improvements in either amplicon-based methods, skims, and capture for getting DNA out of preserved specimens, but it's just trying to make it more routine and enable more labs to do it more easily. So that's the aim of that project. Going from that kind of library construction focus to application, there are a whole series of projects in the kind of meta-barcoding, eDNA, monitoring type work, and this is aimed at improving the pollen forecasts for dealing with asthma and allergies. It's a project coordinated by Simon Creer, but also involving the National Botanic Garden of Wales, essentially airborne grass pollen barcoding. This is a project of ours, but actually done in China, and this is one looking at a diet analysis. It's a medic barcoding project, and this is a very obvious question: what do panda eat? They eat bamboo, but actually they eat about 60 different species of bamboo. Bamboo are very difficult to tell apart. They're taxonomically difficult. They're even more difficult to tell apart when they've been digested by a panda. And this is really about increasing our understanding of panda diet. And there's a whole host of other projects like that, identifying plants to enhance knowledge of animal ecology. There's some neat stuff going on with reindeer grazing on lichens in the Cairngorms National Park, and we're also doing something neat at the minute looking at identifying the plants used by the New Caledonian crows when they build these absolutely fantastic tools for hooking out grubs. And I've just added one fourth project in, and this is related to barcoding. It's at a grant application stage. It's due in about a month's time. We've passed the first hurdle and we're going through to the second stage of this, and this is to completely genome sequence the native vascular plants in the British flora. And there are a number of reasons for doing that, but there is an explicit link through to barcoding here, is that the plant barcodes don't tell all species apart, and as we do complete plastid genomes, often it's the case that those plastid genomes don't track species boundaries, so we just get more data telling us that the plastid genomes don't tell species apart. And ultimately what we need to do is get some data from the nuclear genome, but we're actually sort of just wandering into the dark at the minute because we don't have any real idea about the distribution of taxonomically informative characters across the nuclear genomes of plants. And one of my primary motivating factor is to take the British flora as the Drosophila or the Arabidopsis of flora. It's simple, it's well understood, and use this as a model to better understand how we can improve our knowledge of plant barcodes. And actually as genome sequencing becomes applied to floras and faunas, it's a good, simple case for building up workflows and understanding the practical challenges of doing it. So onto the SWOT analysis. There's some absolutely fantastic natural history collections in the UK, still good taxonomic expertise, and a growing appetite for accessing that material for collection sequencing. There's a lot of interest in barcode applications, and there's some really great individual research groups scattered around the country doing some very neat bits of biology. And just coming back to that bit about the British biota being so well known, it does make it extremely useful when you do have barcoded data sets for linking the sequence data to wider natural history observations and distributions. The weakness I've already mentioned, there's a real, it's proving very difficult to get UK funders to invest in UK barcode library construction, but there does seem to be a lot more interest in biodiversity genomics on that kind of scale, so there's a positive to that. Threats, I've written the B word there, that relates to Britain wandering off into the North Sea away from Europe and leaving behind some extremely good funding opportunities, collaboration opportunities, and actually a lot of uncertainty about what will happen there, and also uncertainty about the access to talent and researchers, and we're really seeing that at the minute. So that makes me very nervous, but let's just be hopeful it will be all right. Opportunities, collections, mining. There are some good funding opportunities coming out in the UK, a lot of money going into the Global Challenge Research Fund, and this is linking science research to the overseas aid budget. So projects that look at addressing the sustainable use of biodiversity and barcoding associated with that, see some good opportunities there. And there are various other bilateral international funding opportunities coming through. And again, just reiterating that final point, that there does seem to be great enthusiasm for natural history genome sequencing type projects and opportunities that come with that. And thank you very much for your time.